Hello, my name is Shubatra Nahenthara, and today I will be presenting a journal titled Raw Transcriptomics Data to Gene-Specific SSRs, a validated free bioinformatics workflow for biologists, published in Scientific Reports on 26 October 2020, authored by a group of Sri Lankan researchers. The contents of my presentation today will encompass the introduction followed by objectives, the methods used in this research, as well as the results, discussion followed by the conclusion. DNA sequencing has evolved over time to encompass first-generation sequencing, followed by next-generation sequencing, and recently third-generation sequencing. Several sequencing instruments have been introduced, each with higher throughput compared to the one preceding it. First-generation sequencing followed by next-generation sequencing as well as third-generation sequencing has led to high throughput and longer reads produced at a lower cost and increased speed. Sequencing and assembly requires a huge quantity of data, not to mention high computational power as well as a long execution time. Approaches in assembly include de novo assembly and reference-based assembly. Applications of sequencing and assembly include the production of molecular markers or simple sequence repeats. Challenges in sequencing and assembly include commercial assembly and annotation tools, which are very expensive. However, an alternative is to use free assembly software, but the problems here or the issues faced are that there are no complete workflows, and it also requires time for the researcher to understand how the software works. Current framework, however, is too complicated and lacks any further downstream analysis. The first objective of this study is to acquire transcriptomic data of S. album or sandalwood from previously published studies and to evaluate the quality of the reads. The second objective is to assemble the transcriptome of S. album RNA sequence dataset. The third, to assess and validate the assembly quality of S. album transcriptome, and the fourth, to design and validate SSR primers for identified genes of S. album. The methods of this research are divided into 4 sections for each objective. Transcriptomic data was acquired from a previously published study through data accession, where raw sequences were downloaded from the NCBI database and converted into FASTQ format using the SRA toolkit. Quality control of the acquired transcriptomic data was done using FastQC, FastX toolkit, as well as Trimomatic software. Figure 1 shows the variation in the number of raw reads in input RNA, and Figure 2 shows the variation in the number of raw reads of the output after filtering was done. Quality control of the raw reads is necessary to filter out any impurities from the sequencing process. Low quality bases and PCR errors might generate reads that aren't completely aligned. The tools here used to evaluate the read quality are FastQC and FastX toolkit, which are able to visualize base quality scores, nucleotide distribution, identify any contamination, and GC bias. Assembly of the transcriptome of S. album RNA sequence dataset was done via de novo assembly using the Trinity de novo assembler software. This software is able to concatenate all forward read files and reverse read files separately for multiple libraries and is able to normalize large datasets. Table 1 shows the summary statistics of sequence data and de novo assembly, where 117.9 gigabytes of raw reads were generated. Over 600 million bases were arranged into 700,000 contigs, and this was further arranged into over 600,000 unigenes, with the N50 value of 659. Trinity is a software which provides high-quality assembly. However, any assembler chosen must suit the purpose of the research. In this case, de novo and transcriptome assembly was done. In the Trinity software, 3 software modules run sequentially on RNA sequence reads, and the software itself is programmed to remove any ambiguous or erroneous reads. In the Trinity software, separate options for single-in and paired-in reads are available. And this software is also capable of normalizing a large dataset to reduce computing requirements, minimize time, and retain sequence coverage. Within a single command with input parameters from the user, normalizing, trimming, and assembly can be done. Three softwares were used to assess and validate the assembly quality of the S-album transcriptome. These three softwares were the Bowtie 2, Busco, and Transrate softwares. Table 2 shows the summary of statistics of Bowtie 2 validation, where a high percentage of proper pairs implies a good quality assembly. Table 3 shows the summary of statistics of Busco validation, where 91.7% of the transcriptome were complete orthologs, and this implies a good quality assembly. Table 4 shows the summary of statistics of the transrate validation, where a low percentage of good mapping was obtained, implying a low quality assembly. The S-album transcriptome assembly was of good quality. Discrepancies, however, in the metrics show further assessment tools are needed for validation of the assembly. Transcript mean length was around the value of the original study from which the transcripts were assembled. However, poor quality assembly should not be used in downstream analysis, as this will result in poor quality analysis. Design and validation of SSR primers were done using BLAST batch primer 3 followed by an oligo analyzer, and the primer was validated via PCR amplification followed by gel electrophoresis. Table 5 shows the SSR motifs and primers designed for the 10 gene sequences of the S-album. Figure 3 shows the PCR amplification of the SSR markers. And figure 4 shows the agarose gel electrophoresis of the SSR amplified products of 3 Sri Lankan album accessions. SSR primer design is a downstream application of transcriptome assembly. It is done to develop gene-specific markers for future breeding efforts. The primers in this research gave clear PCR products, but some with a larger than expected size. This was due to existing biological variations because the original accessions were taken from an Australian accession, but the in-lab RNA sequences were from Sri Lankan accessions. In conclusion, the methodology in this journal was developed to assemble a transcriptome and evaluate the accuracy from raw data using free tools and software. However, tools and software must be always chosen to suit the data needs and the end goals of the research. The markers in this study were amplified successfully, which verified the efficiency of the workflow. The workflow described was also user-friendly, whereby the input required was a raw sequence data and the output received was a draft or a complete transcriptome. The modules can be executed separately in the flow of this research, which allows handling of the intermediate files generated from each step for different types of analysis. Thank you.